Genetics and inheritance mutation, guys. Today we'll be dealing with past examination questions that are related to mutations. So today's date 5 April 2024, and then time there is 36 minutes past 1. So welcome to my class, guys. Sanibonani, dumelani, avsheni, hello, nda varimini minjani bakahina, and then siyaphila. Okay, guys, without being said, let's not waste any of your time, guys. Today's Friday. We know Friday marking the end of week 3, so week 4 we are going to start it next week. Tomorrow, Saturday, I'm not sure if I'm going to send any video, since we will have covered everything. So then another thing, I have Wi-Fi nyana, guys. Maybe tomorrow we can do my live video here on YouTube or on Facebook or anywhere, guys, that we can do. Then let's know each other, guys. We have people from KZN, we have people from Mpumalanga, Free State, Limpopo, Gauteng, all corners of South Africa. So we are here, right? We started at week 18. This week we started at 18, and then we are here, guys, Friday. So previously on our African transcribers, we did concept of inheritance, monohybrid crosses, sex determination. We did past examination papers, and then we did dihybrid and blood grouping. We did genetic lineages, pedigree diagrams, and then we did mutation. You still remember our plan, like Monday, Tuesday, Wednesday, Thursday, Friday. So Friday we said that we are going to do mutation, and we are also going questions that are related to mutation, since we did mutation yesterday. If you missed my previous video, guys, go back and watch it. Then today we'll be familiarizing ourselves with the questions that they ask us a mutation, right? Okay, so today this week we learn how to interpret pedigree. You'll agree with me, and also the other guy sent me the pedigree of a worksheet of him interpreting this pedigree, and then we did past examination paper pedigree, and now we'll be doing the mutation. You'll see how they ask this mutation, right? So next week we'll do genetic engineering, genetic paternity testing, where we use DNA, and then we use genetic links, right? We link mitochondrial DNA. So with that being said, guys, let's move on. Let's not waste much of our time, right? We did mutation. If you can bring it down, Yana, for a revision, small revision, we did mutation, and we defined mutation previously, right? So we said a mutation is a sudden change in a structure. It's a sudden change in a structure. Sudden change in a structure of a gene, structure of a gene in an organism. There's change in the structure, there's change in the genetic makeup. You can say this is the change in the genetic makeup or genetic composition. We talked about this, right? We are just passing by, and then we said the effects of mutation. It's either the mutation is harmful, it's harmless, or it's useful, right? Then we talked about this. When a new protein is being produced, we can say it's harmful. When the mutation is taking place at a non-coding part or non-coding portion of gene, of a DNA, sorry to say, of a DNA, we can say that a mutation is harmless. And then when we are having a useful mutation, for an example, if maybe the mutation is going to be a survival, is going to contribute on an organism survival chances, we say that is the useful mutation. You still remember that? And then we talked about mutation as it contributes to genetic variation, and then we did an example here, crossing over, due to random assortment. That was the two reasons why we were talking about genetic variation, and now we are adding that also we find that this mutation can also contribute to genetic variation, right? And then from there we did a definition of gene mutation and chromosomal mutation. I told you that there's a difference between when they ask you define mutation, define gene mutation, define chromosomal mutation. There's a difference, right? A gene mutation is a mutation that is happening on a genetically level, or on a gene level, or on a nitrogenous, we say on a nitrogenous basis, right? In DNA, nitrogenous basis in DNA, or some they say nitrogenous basis or nucleotides in DNA, right? So when the changes is taking place inside the DNA or on DNA itself, we say that is a gene mutation. And then chromosomal mutation, when the change is taking place on a structure of a chromosome or the number of chromosome, we refer that as a chromosomal mutation or chromosomal aberration. You still remember that? And then we move on. We said we have two types of mutation. We have gene mutation and a chromosomal mutation, right? And then these two mutation, they can lead to a genetic disorder. So the question can come that name the genetic disorder that is being produced, or that is being that is the result of that genetic mutation. You know the disorder. We are talking about hemophilia, color blindness, or blood Down syndrome. And we also say that we also have sickle cell, and we also have cystic fibrosis here, right? So when you understand or you pay much attention to hemophilia and color blindness, you can also bring another question, but the questions will be like this. And then Down syndrome here, when we are dealing with chromosomal aberration or chromosomal mutation, right? When one chromosome can be like this and then the other one like this, because here we had a change in the structure, we say this is a chromosomal mutation. Or if we have 3 chromosomes instead of 2, or we have 1 instead of 2, we means that that is also a chromosomal mutation. It's a sudden change, right, in a genetical structure or a genetical makeup, right? And then trisomy, we did this. And then the process called non-disjunction, right? This is the result of non-disjunction during meiosis. So we did this. We can move on. That was a small few 5 minutes revision of what we did previously. So you can agree with me. We can move on. Hemophilia, color blindness. As much as I'll feel like I can explain this and over again, guys, we'll have to move on at some point. And then here yesterday we were trying to understand deeply on our mutation using our green book, right? Mind the gap, that this mutation can be caused by X-ray or can be caused by a certain chemicals. We didn't talk about it previously. We didn't talk about it on the examination guideline, right? So it can be natural. You still remember I said this, it can be natural where this non-disjunction is happening, or it can be caused by X-ray, where you find that this X-ray, because they are rays, they can go and they disturb the formation of a new strand during DNA replication, right? A certain chemical. And then we talked about harmful, harmless, and a useful mutation. You still remember that? Today we are just going to familiarize ourselves with questions that they bring when they are asking us a mutation based question, right? So something like this, I told you yesterday, or I asked you yesterday to go and do this activity as your homework. Some of you maybe you did, some of you didn't do. It's fine, guys. We welcome all of you. Life science, so we can come and say correction here. Allow me to corrections, and today's date 05/04/2024. So thank you. Activity, this was activity number activity week number 3. It's fine. And then the activity was like this. They said a sickle cell disease, a sickle, you are doing correction, you will mark yourself, right? A sickle cell disease is caused by a recessive allele. It's caused by a recessive allele and first appeared in humans as a result of a gene mutation. So this sickle cell diseases, it was caused by a gene mutation. But the table below shows the number of children born with sickle cell diseases in some regions in a particular year, right? So these are the number of children born with sickle cell, and this is the region, right? So we go and study the table, and then and this will be before the question, right? It's just that here they place them together so that we will have a table and the question at the same strict. And then number 1, they will ask us, they asked what is a gene mutation. So you see, because they talked about gene mutation, now they ask you a gene mutation. They can ask you what is gene mutation, give 2 types of gene mutations, or give disorders that are linked to gene mutation. And then since they said sickle cells, you know, we have sickle cells, we talked about them yesterday. We have albinisms, we have hemophilia, we have color blindness, we have all those gene mutations that we talked about them yesterday. So if you missed, go back, guys. If you are not even sure what this correction is about, go back. So but what is a gene mutation? A gene mutation, by this time you should be able to understand, or you should be able to define that a gene mutation is gene mutation is a change, you can say is, or a change, is a change in the sequence, change in the sequence, change in the sequence of nitrogenous bases, or nucleotides in DNA. So here's the thing. But what is a gene mutation? A gene mutation is a change in the sequence of nitrogenous bases. These nitrogenous bases, they are changing, right? Or the nucleotides. What are they saying by this? You know that there's a difference between nitrogenous bases and the name nucleotides, right? The base, it's everything there. Then the nucleotides are the guanine and the thymine. They can say that the guanine and what they changed, something like that, right? So that will be your 2 marks. This will be your 2 marks, where now being able to say that this is a change in the sequence. The change in the sequence, you'll have 1, right? Change in the sequence of a nitrogenous base in DNA, you'll have 2. For you to say that this change is happening in a DNA, you'll have 1, and then what is happening in DNA, there is a change in a sequence, you'll have another 1. So I hope you understand where this 2 marks is coming from. And then number 2, which region had the highest number of children born with sickle cell diseases in the year, right? So we go to our table, we see that the total one, it's here, it's 503,733, and then we are going to see with the highest number. They say the highest number, we're going to look for highest number, and yes, the highest number, and then the highest number is Nigeria. So when now you'll say Nigeria, then you get your 1. Then we move on. Number 3, what percentage of the worldwide total of children born with sickle cell diseases came from the Democratic Republic of Congo? Show all calculation. And then here I also like to say something, that when they ask you what is the percentage, for you to calculate that percentage, the moment you multiply a certain answer with 100, that is your answer, guys. That you see, you can see as 3 marks, right? So this, they can give you this answer because you understand that we have a fraction, and that fraction is being multiplied by 100 for us to get a total percentage, right? You agree with me that? So now you will have another way is the other mark is going. The other mark will be the total, and the other mark will be your division line here, the amount that you divide. So they want the disease came from Democratic Republic of Congo. We go to the diagram and we see, yes, the Democratic Republic of Congo, and the number who were born with sickle cell, they were 39, they were 746 divided by the total, because we want the percentage of all worldwide, right? So we'll divide it by 305,733. So this will be your answer there. And then the answer here is something like a calculator. You say 39,746 multiply by you see 39,746 divide by 305,733. I don't know, I want my calculator to be just small. Yeah, so I want my calculator to be like this. Fine. Equals 2, and then I thought maybe I canceled this. Sorry guys, sorry, bear with me. 39,746 divided by 305,733, then equals 2. Then the answer here multiplied by 100, you see that is going to be 13%, right? It's fine. Then 13.00. So that will be our answer. Our answer is going to be 13%. You see, I did something and my thing now, it's not working properly. Okay, let's move on. Then you get another 1 mark there for 13%. And then we come to number 4, and then number 4, it says use the letter D and the small letter d to give the genotype of a person who suffers from a sickle cell diseases, because we know that this sickle cell from our line here, they said is caused by a recessive allele, right? So since it's caused by a recessive allele, for someone to suffer that sickle cell has to have a small d and a small d. You'll agree with me, right? Because it's a recessive alleles and for them to be phenotypically visible they have to be 2, right? And then number B, but give us the genotype of a person who carries the allele but does not suffer from the disease. So the answer will be D and small letter D. Then you have your 1, you'll have your 1, then total will be 1 2 3 4 5 5 6 7 8, right? It will be 8 out of 8, then that will be your 8 out of 8. Then when you'll comment how much you got. 04 05, sorry. Then you'll comment how much you got from this, right? So let's move on. My thing is no longer working properly in this page, and then we move on. We go to our question papers, right? Question papers 2021, 2022 and 2023. You still remember this question paper here? 2021, 2022, 2023. We did the scan. We even scanned the question paper. Everything, it is fine. We did this question here, if you still remember. And then we did this also. We also did this previously when we did the questions regarding a pedigree. And then now we are going to do 2.4, and I saw another, this was not the paper, I'll show you. So okay, it's fine. Let me see if my thing, yeah, it's writing properly. Okay, it's fine. They said read the extract below. Read the extract below, and you should read and understand. Yeah, they should just say read and understand, because if you don't understand this extract, guys, you are going to have a problem answering here, unless with these questions that there are general answers, but the one that will be requiring you to extract those questions from this will have some problem if you don't understand. Let's go and read it together. Okay. Researchers have discovered that members of the researchers have discovered that members of a particular family have high bone density that may be caused by a gene mutation, right? So they are thinking, or it may be caused, they are just thinking it may be caused by a gene mutation, since some of these family members, they have high bone density, right? And then high bone density reduces the risk of bone fracture. So as you can see, this high bone density reduces the risk of a bone structure. It means that this mutation will be a useful mutation. It will be useful, right? And then let's go to another paragraph. It says 20 members of a family had their bone density measured. So they measured the bone density, and the DNA sample was taken. So they measure their bone density, and they take the two things. They measure their bones, and then they take their DNA sample. That 7 had high bone density. So we have 7 of them out of 20 that they had a bone density, and then the remaining one will be 13 out of 20. Yes. And then the high bone density occurred throughout their bodies, but especially in the spine and the hips. So you find that this high body density is spread out through the body. Like you can even find it on arms bone, but they say here we find them more on spine and hips. So 2.6.1 is the question, but from the extract, okay, identify 2 areas in the body where bone density can mainly be measured, where they are being accessively, so that we can measure them. If you can go to our extract, I don't want to say much, but if we can go to our extract, the sentence where they were talking about the body, what is this, right? The last line, he said the high bone density occurred throughout their bodies, but especially in the spine and the hips. So this spine and the hips, that's where we are going to be able to measure this bone density, spine and hips. So if we are going to, we can measure it everywhere, but because it is even more especially on the spine and the hips, that's where we can mainly measure this bone density. And then 2.6.2, and then describe what a term mutation is. Describe what a gene mutation is. Yes, another question is coming again. So if we were doing with chromosomal mutation here, they were going to ask you describe what a chromosomal mutation is. Because now we are dealing with a gene mutation here, for sure they will ask you what is that gene mutation. And then when you say a gene mutation, you know a gene mutation is a change. A gene mutation is a change in the sequence change in the sequence of the nitrogenous bases in DNA, or nitrogenous bases or nucleotides, right? Nucleotides in the DNA, right? So that was it. Describe what a gene mutation is. You are describing it, or you can put the mutation. This is a change that happens inside a gene of DNA, right? You can get one there, or you can get two. I'm not sure how they can place these two, but if you are describing what is a gene mutation, for sure you'll have to say there's a change. You also have to say the sequence. You also have to give us that where is this mutation taking place. Is taking place inside the DNA, and what are those things that are changing inside the DNA to say that a mutation is happening inside the gene, the nitrogenous bases. So you see. Then 2.6.3, explain why it was necessary for researchers to collect DNA sample. So we see that they did two things. They firstly measured the bone density, and after measuring the bone density, they took their DNA samples. So what was the reason? Here they want us to give them a reason why they were taking this DNA sample. Here's the reason. Because they were you see, here they said they have a high bone density that may be caused by a gene mutation. The reason why they took their DNA, it was to study, you understand, it was to study their DNA structure, their DNA sequences. It was to study their DNA sequences to find or to check if high bone density it is the result of gene mutation. Do you understand? Because they found 7. If they can go to these 7 of them, and then they studied their DNA. After studying their DNA, they find that this DNA is different from this DNA this side. It means that we had a mutation. Maybe some nucleotides were added, and when these nucleotides were added, they went to the bone density and increase its density. Going to the bone and increase that bone density. Or we can say, because this one they have a normal bone density, and this one they have high bone density, and we find that no, this DNA is fine, but in this DNA there's something that is missing. It means that the mutation, maybe they were just taking the DNA sample to check the DNA structure, to check if the mutation is there. You understand? To understand or to study their DNA to see. It's 2 marks. If you can say this, to study their DNA sequence, they're taking it to study it, right? To conclude whether a gene mutation is one that is causing high bone density. So you see how we interpret this instruct, right? The answer has to be here. The answer that you are giving here has to be corresponding with what you were told here. Then 2.6.4, state the effect of this mutation. State the effect of this mutation. You still remember that yesterday we talked about the effects of mutation, right? We said it's harmless, it's harmful, it's useless, right? So here, when they say this question, state the effect of this mutation, someone might come and say a useful mutation, or harmless or harmful mutation, but you have to not forget that in life science, when we ask you the effect of something, it's like we are asking you the consequences of this mutation. If when you have this mutation, what is it that you are going to be affected on? Do you understand? If when you have this mutation, what is it that you are going to be suffering from? Do you understand? So in this scenario, there's no suffering, there's just a benefit. So the effect of this mutation, it will be high. This mutation will result in high bone density. What is the results? What is the outcome of you having this mutation? You will have high bone density, or you can say you can reduce or reduced risk of bone fracture, right? Bone fractures. So what if you have this mutation is going to affect you in a positive way, right? This will be a positive effect. So this is going to affect you in a positive way because it's going to reduce the risk of your bone being fracturing, being broken, right? And also you can just say it's 1 mark. You can also just come and say this one will increase the bone density, right? So 2.6.5, let's go to 2.6.5. It says calculate the percentage of a family. Calculate the percentage of the family members who had normal bone density. We calculated that. We found that 7 of them out of 20 they have, and then we also, it means that 13 out of 20 they are normal. They have a normal bone density. Show all your calculation, right? 3 marks, 3 marks. I'll first show you the first calculation. Or first calculation, you can come and say 7 at the one should be straightforward. You are going to say 13 out of 20 multiplied by 100 equals to, right? So let's go to our calculation. Let's go to our calculation. We want 13 divided by 20 multiplied by 100 equals to 65%, right? So it's equals to 65%. So when now you can get 1, 2, 3. As you can see here, they even gave you that 1 year multiplied by 100. So let's say when now you have another way to come up with the 65%, right? You are going to say, we know that we were told that in an extract that the 7 of them out of 20 they had a high bone density. So if we can find percentage of this, we are going to have 35%, because we are, you understand, right? This doesn't need a calculator. We are going to have 35%, and then that 35% I'm going to subtract it out of, I'm going to subtract it out of 100, right? Then 35 here, 35%, and then it will be, we will have 65%. So this one will be our final answer. Then these 3 marks they are going to give you this one here. Do you understand? This one here of multiply by, because they said calculate the percentage, you being under, you having that, I've already said this, you having an idea of that we need to multiply our fraction 100%, so that will have a percentage. It means that you understand the question. Will give you this one, and where's the other one there, where's the other 2 coming from? Because this one is wrong, this one is wrong, we are going to give you 1 here, and we are going to give you 1 here. So you see where's the 3 came from, and we also 3, you see where's the 3 came, where's the 3 coming from here, right? So it's not like they will say we want to see 13 out of 20. If that person had 13 out of 20 in his answer, that person will have to mark that person. No, we might see a different answer here. We might see, maybe you might say, hey, let's say 10 divided by 20 multiplied by 100, and then equals to, and then you find the answer, and then you come and say what times 100 equals to what times 100 equals to what. At the end of the day, you said this one will be 65%, this one will be your answer. It means that you give her the rightful answer, and you know that we were multiplying by 100, and then whatever you got here, if it's understandable, we'll give you another 1 for your calculation, because they say show all your calculation, right? So I hope you understand that. Let's move on to another question, right? And then there's another question, 2021, 2022, as we have promised, 2021, 2022, and 2023. And then here will be 2022. We also did this. We even did this, guys, if you still remember. And then from there we are coming to 1.3, right? As you can see at 1.3, but the diagram below represent the part of an abnormal, I would like to use red, right? The diagram below represent part of an abnormal human karyotype, right? So this one is an abnormal karyotype. So when now with your understanding of a human karyotype, you will see that this had to be 222. We should be having 222. So at a chromosome number 21, we have extra chromosome, and you know what is happening when we have this extra chromosome. So you already have answer. And another here you can see we have a short MBB here. It means that, you understand, when now you have an answer, because you can interpret the karyotype, right? After interpreting the karyotype, you can go to your questions now. 1.5.1, they said how many autosomes are shown in the diagram. How many autosomes? So when now you know by this time that we have autosomes and gonosomes, and since 23 it's our gonosomes, it means that they want the number of these autosomes, right? How many autosomes are shown? It will be 1, 2, 3, 4, 5. That will be only 5 autosomes, right? You know what is autosomes, you know what is gonosomes. This is an autosomal. Then maybe they can ask you, because they said how many autosomes are shown, they don't want autosomes or from 1 up until 22. You can come and say there will be 20, there will be 40, because there's an other one, minus 2, 40, 46, 47, minus 2, 44, right? Something like that. So 1.5.2, Barry, name the type of chromosomes represented by pair 23. Name the type of chromosomes. This will be a sex chromosomes, right? Chromosomes. These are the type of chromosomes represented here. We are representing a sexed chromosome, or you can just say gonosomes. I don't think they can penalize you, gonosomes, right? I don't think they can penalize you, because the gonosomes are types of chromosomes at pair number 23. These are the sex chromosomes, right? And then 1.4.3, 1.5.3, sorry to say, as KISS, they said name the disorder represented in a diagram. Now you know there's a difference between disorder. Pay attention here. Name the disorder. They can say name the mutation. They can say name the mutation represented in a diagram. When you will come and say chromosomal mutation, but now they want you the disorder. Yes, they understand you it's a chromosomal mutation, but give us a disorder represented there. And disorder represented there, we know with 21 we can say trisomy or Down syndrome, or both of them, Down syndrome or trisomy 21, right? So anyhow, we'll know that you know what is the disorder is being called, right? Number B, name we are still at name the process during anaphase of meiosis that result in the abnormal number of chromosomes in this karyotype. As for this time, you should be understanding that from this IPMAT thing, interphase, prophase, metaphase, anaphase, telophase, anaphase, we are pulling them aside. So because they don't separate here, this is the process that results in having this chromosome. What the process is called non-disjunction, right? Non-separation. I'm not sure if you write non-separation they can give you, but non-disjunction will be the rightful answer there for them to just give you that one. And another thing, 1.5.4, they said state the gender of the person represented in this karyotype. They want a gender of the person. If you can go up and still, you know that we have X and X, this one a female, and then we have X and Y, this one a male. You see the X one is this X here, right? This is where the X is coming from, and then the Y one, this is the Y is coming from, right? So now you see where's the X and Y coming from, right? So if this was a female, the person was going to be an X and X. If you don't know this by this time, you want you need a prayer, really prayer. We are in a real trouble. Okay, so let's move on, guys. You know that this one is a female, this one is a male. When now you'll come here and write your male, the gender, you'll write your male, male, and then you get these 5 marks, guys. It was only 5 marks, 5 sex chromosome, Down syndrome, non-disjunction, and a male figure, right? So I'm not sure if this was the question paper. Yeah, this was the question paper that I also saw another question here. This question, this side, 2.2, same question paper. And then 2.2, they said a mutation has occurred on a section of an mRNA. So there was a mutation that occurred in a mRNA. So here they brought 2 questions, a mutation, 8 marks. So 5 plus, it will be 5 plus 8, 13, right? You'll have your 13 placed aside. So but a mutation has occurred on a section of an mRNA molecule as shown below. So I've told you that this mutation, this gene mutation, let's start that this gene mutation, it can happen during DNA replication, right? And then when that DNA replication, we can have an mRNA that have incorrect sequence, right? Or we can have this gene mutation during DNA protein synthesis, right? DNA transcription, right, or DNA replication. So when this happening in mRNA, we can have an original sequence. This is this thing, original sequence. The mutation here is happening at at an mRNA, right? So it means that we have an original sequence of an mRNA that were supposed to be produced, but something happened and then we resulted in a mutated sequence. Let's see. They said this one was supposed to be this three was supposed to be AUG. You know that we read them in codons, right? 333. AUG, it was fine. GAA, it was fine. AUA, it was fine. CCG, CUG. So this codon here, it was mutated, right? CCA, GGA. So you can see that this codon, this new mRNA, it's carrying a codon that's going to code for a different amino acid, right? That's what we can think, or that's what we can now be a hypothesis, not conclusion. Conclusion, we are going to find it here. If this CCG, let's see, where is CCG? There's CCG. CCG, it code for proline, and let's see CUG, it code for leucine. So it means that a protein, a amino acid was changed there, right? So we know that in a protein, if one amino acid is changed, we are going to affect the production of that protein. That protein can end up being partially or non-completely functional, right? So another thing, if for example, if this CCG, which was coding for proline, was changed by CAA, the proline, it was still going to be proline at that amino acid. So the protein, there was not going to be any change to a protein. So you see, same as here, they only gave us only this. You understand by this time that this amino acid, because we only have 20 amino acid, we are going to have a codons that are repeating, something like this, that are repeating but coding for the same amino acid. So it means that if this can mutate to this, or if this can mutate to this, the change, it won't be there, or we won't see the change in our protein, right? So the mutation will be harmless, right? So let's go to the question that I was just trying to explain, because it's something that we also have to make you understand, guys. So thank you, guys, for those who are understanding. 2.2.1, name the type of mutation that has occurred. Name the type of mutation that has occurred. When now you are going to say gene mutation. Here's the thing. There are those of you who want to act like you know things. Someone was coming to say, because C, it was replaced with U, this was a point mutation. It's what I told you that the point mutation and point shift mutation, we are not going to entertain that in our learning, right? Although we can come and say this one was a point mutation because one chromosome was replaced with the other, right? This was the replacing the gene point mutation, but here they only want you to say this is the gene mutation because it's happening on a gene level, or it's a chromosomal mutation. The time they will ask you the type of mutation, they will want you to give either gene mutation or chromosomal mutation, right? I hope you understand that. So 2.2.2, it says give a reason for your answer to question 2.2.1. Why are you saying this is a gene mutation? It's a gene mutation because the codon, or because the sequence of a gene, you see the sequence, it is because the sequence was changed, or there was a change, the sequence changed, from CCG to CUG. 1 mark. The only just 1 mark. We know that since it's a gene mutation, with the one that deals with gene mutation, we are dealing with changes in a sequence, or the nitrogenous base. You can say there's a change in a sequence, or there's a change in a nitrogenous basis, or the nitrogenous base here changes. Nitrogenous basis here in the mRNA changes from this to this, right? 1 mark. And then 2.2.4, we can come to this one, but the table below shows some mRNA codons and the amino acids that they code for, right? So these are the mRNA codon. On some questions, they can just give you the codon like the previous homework that It's not the previous homework. They can just give you the codons, and then when now you know that mRNA is dealing with the codons, or they can give you a tRNA anticodons, right? Anticodons. So you know that if you have a T, this one, for example, let's use this one. This one is GGGA. So it means that the tRNA anticodon will be CC uracil, right? But both of them, they will be bringing the glycine. This mRNA is going to produce glycine, or the tRNA that's going to bind with this, it's going to be carrying glycine, right? So here they give you the full names. You can find that in some books. They just methionine, they can just MET, and then leucine, they can this glycine, they can say GLY. So when now you write what they given you. If they given you methionine, you write the full name methionine. If they gave you the abbreviations for proline, they can say PRL. Then when now you'll just write these abbreviations, right? I hope you understand that. So let's go to our question. Number 2, it says state the number. Okay, number 2, state the number of different amino acids coded for by the original sequence of mRNA molecule given above. So when we want the different amino acids, right? So you know that this amino acid, one amino acid, it's been carried by a tRNA having 3 anticodons. So if we are having 3 codons on an mRNA, that 3 codons, they result to 1 amino acid. So here they want us the number of different amino acid coded for by the original sequence of the mRNA. So we go up and say 1 this, 2 this, 3 this, 4 this, 5 this, 6 this. Right, 1 2 3 4 5 6. So when now you'll come and say, Barry, state the number of different amino acid coded for by the original sequence of mRNA molecule given above, right? So let me see something. AUG is methionine. I'm not sure if they want these 6. I mean, say the number of different amino acids. Different amino acids, CCG. I want to say something, CCA. You see here we have a same amino acid, right? Here we have the same amino acid, CCA. It's also same as CCG. So since they're saying state the number of different amino acid, so this different amino acid coded for the original one, so it means that it's going to code for 1 2 3 4 5, right? Since it's 1 2 3 4 5, right? GGA is going to code for glycine, and it's also going to code for CCA. CCA is going to code for CCA proline, and then CCG also proline, another different, and then AUA is going to code for isoline. GAA is going to code for glutamic acid, and then AUG is going to code for methionine. So it will be 5, right? This will be 5 different amino acids that will be coded, the different amino acid, when these two code the same amino acid, right? So we move to number 5. Number 5, it says give the anticodon. Give the anticodon. Here we are talking tRNA, right? tRNA. Give the anticodon. Give the anticodon of the tRNA molecule that carries the amino acid isoleucine. Where is isoleucine? Number 1 here. This is isoleucine, and then the mRNA codon, it's AUA. So we want the anticodon that will come and join this AUA from tRNA. You know it doesn't have thymine, so you are going to say uracil, adenine, uracil, ne? Uracil, adenine, uracil. This will be your anticodon. Then you get your 1 here, right? Then codons, they match with anticodons and they carry the iso, something like this, right? So number C, use information in the table to describe the effect of the mutation on the protein formed. Use the 4 marks. Use the information in the table. Which table? This one, right? To describe the effect of the mutation on the protein formed. Okay, the protein formed, it means that you are going to say UC CCG coding for CCG coding for proline. This we are using this the CCG coding proline was changed was replaced by CUG coding for coding for leucine, right? So with these two, this result in different protein structure resulting in, but use the information in the table to describe the effect of the mutation. We want the effect of the mutation on the protein formed. Since this is resulting in a different protein, it means that the protein will be or can be affected, or I want to incorporate something that you'll understand, but we'll say the protein, yes, it will result in a different protein, but we also want to incorporate this protein that will be formed will be partially is it partially or non-functional. So this is the effect. How is this going to affect this protein? The protein is going to this one is going to produce a protein that is partially or non-functional because the proline was replaced with a leucine, right? And the results in the protein structure, the protein structure will change, right? So that was your 4 marks. Your 4 mark will be as telling that this CG was coding for proline, and that CUG was coding for leucine. And then when this happening, we are going to see a different protein structure. Maybe like it was going to be like this, but since now it's changed, it means that we are going to see something like this. It means that their protein structure is going to change. With the change in the protein structure, you know that we are going to affect the protein functionality. It's going to be partially or non completely functional, right? So that will be your answer here, 4 marks, 3 and then 4, right? So from there we can go to 2023. You can see now I'm chasing time. We can go to 2023. This was our 2023 question paper. You still remember it, right? You still remember we did those. We did the question 2.3, right? So 2.3, read the information below and understand. You know, right? You read the information and understand it. A gene, let's read it together. A gene, a VKORC1, a gene VKORC1 code for a blood clotting factor in a humans. So it means that when you don't have, when you are suffering from hemophilia, still remember hemophilia, you have a dysnormal that this gene here is not producing a blood clotting factors, right? And then you result in hemophilia, if you still remember that. And then this gene is made, but when you don't have, when you read this, you don't have to have a like the information that you can link the information with, right? I'm just bringing this hemophilia thing because it's familiar with blood clotting factor. But the gene is made up of 163 amino acid. So this amino acid, they are like joined like this up until 163, and this one are joined by a peptide bond, you still remember, and then one amino acid there was brought by a one tRNA. You remember that, right? So we join them when they are 163. That's when now they are going to produce a blood clotting factor, a protein, right? So let's move on. And then it says a mutation occurred that affected amino acid 121 and 128 and 139. So let's say maybe this amino acid was affected and this amino acid here was affected. We'll say this one, it was 128 and this one was 139, and then we also have another here, right? So this one was affected. You can say that the mutation affected this amino acid, but the sequence CTG, you see now the sequence CTG changed to CAG and the TAT became TCT. So the sequence change, it was CTG was changed to CAG. This was changed to this, and we also changed TAT was changed to TCT, right? So this affected the amino acid. So it's either amino acid changed since there was an effect in this amino acid. It means that the amino acid changed, right? That's what we can get from this. But this mutation has been transmitted as a autosomal dominant characteristics through the generation. So it means that probability of you being affected by this are high, because if it's caused by a dominant, a recessive over a recessive one, when you are like this, you understand, you'll be affected, and then you'll need small letter d, if we are using small letter d as a resistance to have this blood clotting factors functioning well, right? Because mutation caused this. So, and then this, it was transmitted. We can move on from there. And then where the mutation has resulted in resistance to warfarin drugs in human. So resistance, you know this, if we are saying something is resistant to something, it means that when you drink these drugs, or when you take up these drugs, they are now inhibited. They cannot work best to treat whatever, or to treat these diseases. Or it caused that when you drink this blood here, when you drink this drug here, the drug won't have effect, right? And then what is the reason of taking this warfarin drugs? But warfarin drug is used to treat it's used in the treatment of thrombosis. Thrombosis results in a formation of blood clot in the artery. And then warfarin causes the thinning of blood to break down the blood clot. So here's the thing: if you are saying you are suffering from this thrombosis thing, it means that your blood is going to clot, right? Your blood, you are suffering from a blood clotting on the arteries, right? So when you have this, we give you warfarin, and when we give you this warfarin drug, this drug is going to thinning, or it's going to make the blood group to be thin, and then it will break down the blood clot. It's a lump, something like a pap. You understand? When you have a pap and then you clot it like that. So this thing is going to clot this. But if now you are being resistant to this warfarin, it means that when you have this mutation from this statement, you can see that it becomes resistant to warfarin. It means when you take this warfarin, instead of this warfarin to come and thin the blood, your blood now find out that it cannot respond to warfarin drugs. It means that these drugs cannot come and do its work. It cannot come and thin the blood so that your thrombosis will be reduced. So let's go to our questions. That was just an explanation so that you'll understand the what is they are being saying. Yeah? That's what you have to do when you read this extract. You'll have to read them to understand what is happening. What is it that they're telling us? They are not telling us stories here. They are telling us an information that we are going to use to answer these questions. So number 2.3.1, they said give one piece of evidence from the information that shows that the mutation of this gene occurred in a DNA molecule. Where are we going to find that? We are going to find it the sequence change, right? The sequence change. When the sequence was changing, it means that we were having a gene mutation. But the sequence that was changing, they were changing CTG. You can say CTG changed. CTG changed to CAG. Therefore, the molecule was a DNA because we see thymine being involved. So here's the thing, right? So because you know the difference between DNA, if it was not happening on a DNA, it was going to be happening on RNA, right? So because it is happening, you know the difference between DNA and RNA. DNA contains thymine, and then RNA, we see them by containing uracil. Since we are seeing TT here and TT here, it suggests or it concludes that this change is happening at a DNA level, right? That's what you are going to give it. So just a 1 mark. Now how you'll say it, it will be up to you. You can say thymine, DNA, yeah. I will explain on your understanding, but we want to see this thymine being linked to DNA, right? So 2.3.2, they said how many nitrogenous bases code for VORC1, VROC1 gene, right? How many nitrogenous bases? How many nitrogenous bases? Nitrogenous bases G, T, C, U. How many of them code for this? So if we can go to our extract or to our information, we can see that this gene is made up of 164 amino acid. It's made up of this 164 amino acid, and we know that one amino acid, it will need 3 codons from mRNA to be something like this. This one mRNA here, it will need 3 anticodon and 3 codons from mRNA for it to be placed there. So since we have 163 of this, it means that we had 1, 2, 3, we had multiplied this by 3 to give us an answer, right? We'll multiply this by 3. We have 333 of, we have boma 333 by 163. Let's go and write the answer. We are going to say 163 multiplied by 3, because 3 nitrogenous base, they are going to result to 1 amino acid. Then we go to our calculator first 163 multiply by 3, and then that will be 489. That will be 489. Then your 2 marks, 1 mark, 1 mark here. Sorry about this. 1 mark, 1 mark here. Then 2.3.3. Describe what is meant by an autosomal dominant allele. But 3 marks, right? So when you'll have to give us, to describe what is meant by this, you'll have to make us understand what are they saying when they are saying a autosomal dominant allele. When you'll come and say this is an allele, let's write it here. This will be an allele. This will be a dominant allele. You can say that, you can start there. This will be a dominant allele. Dominant allele, situated or located outside of sex chromosomes. Or you can say this is a dominant allele not located inside sex chromosome. Whatever way that you'll put this dominant allele and sex chromosome together, that will be your first point. And then another thing, you'll come and say, because they're saying you have answered an autosomal, right? An autosomal is something that is not connected with a sexed chromosome, right? And you have answered that this is a dominant. And then you can also come and say, since it's dominant, it will mask this. It will mask the recessive allele, allele found allele when, you can say it can mask this recessive allele when found in heterozygous condition, right? Condition. Or when you can come and say it will be phenotypically shown, right? It will be phenotypically shown when it is found on a heterozygous condition. Same statement with this one. So when you can get one of understanding that this one is a dominant allele, and this dominant allele is going to mask, or it's going to be expressed when it is grouped, or when it's found in heterozygous with a recessive allele, right? And then you can also come and get the one that you understand that this dominant allele is doing that, but it is not on a sex chromosome, right? That's what they are meaning by that autosomal dominant allele. They mean that this allele is dominant, but it's not situated on a sex chromosome, right? And this allele, if it is matched with a recessive allele, it will be phenotypically shown, right? And then that situation is a heterozygous situation or heterozygous condition. So we can move on to 2.3.2, 2.3.4 there, and then 2.3.4 says the table below shows the amino acids and their corresponding codons. Where do we find codons? We find codons from mRNA. So we have mRNA sequence and we have the amino acid. As you can see here, I was saying that in some question they can give you the the abbreviation leucine, and then some they can give you a full name, something like this, from not leucine, serine, treo, what glycine, those amino acid, right? So whatever they give you, if they give you like this, just come and write the abbreviation like this. If they give you a full name, you can come and give the full name, but you will not be required to know these names. So number A, but explain, right? Number 8 says explain how the mutation on the VORC1 gene result in a resistance to this. Explain. Just explain to us why the mutation in this gene result in a resistance of what, right? So you know that when you explain, you are going to explain that because amino acid were changed, right? CTG there, CTG there, we have it like this side. You can come and see this side. CTG, it was going to code for this one. It's in DNA, right? Another thing, don't forget, we have a DNA. It means that we're going to have CTG from DNA, and it's going to have an mRNA. It's going to be CTG. Our 1 hour mark is up, that my alarm is ringing, but I'll use another, let me use another 10 minutes so that we'll answer this, right? And we will be fine. CTG is going to be mRNA. It's going to code with G, adenine, and cytosine. And this was going to code, we come here to the mRNA codon and see GAC. This is GAC. It was going to code for LEU, right? So this was changed to, you can be writing, this was changed to CAG, which it was going to be mRNA. It was going to be G uracil cytosine. So this was going to code for G. Let's go here and say GUC. This is GUC. It's going to cause GLN. So leucine was changed or was replaced by, or 128. You can say amino acid 28 was replaced, leucine was replaced with this, and then we can go to another amino acid that was changed again, T80. It was going to code for T80. It was going to code for adenine, uracil, and then adenine. And this was going to come here and see our codon. This is AUA. It was going to code for TRY. Then another code was TCT. mRNA, it was going to be thymine. It's going to go with adenine, and then cytosine, guanine, and then thymine, adenine. Then we go to the mRNA sequence and see AGA. Where is AGA? It was going to form ARG. So this was replaced to this. It was 139, if I'm not mistaken. And then with this now, it's 5 marks, and with this now you can come and say, because 128, because in 128 leucine was changed to GLN, and then in 139 TRY was changed with RG. This will result in a different protein. This will result in a different protein structure. Right? So this will also result in a non-functional, or this can result to non-functional or non-partially protein. This will result to when this pharylene, this, because of the protein structure being different, it's going to make it difficult for warfarin to respond. It's going to make it difficult for a human to respond to warfarin, since warfarin will not be able to attach to the protein being produced, right? Because this mutation here, it will be this protein here, it will be mutated, right? Mutated, it means that we'll have a new protein, and this protein is going to make it difficult for a warfarin to attach, so that it will break down, so that it will thinning the blood and break down the blood clot, right? So that was 5 marks there. When I should be explaining this, you can get 3 here, or 2, 3, let's say 3 here, 1, 2, 3, and then 2 here, another 2. You'll get it from here when you are telling us, or showing us how the mutation, it can result in the resistance, right? You have to tell us the protein structure will change, and then due to protein structure changing, it will affect the warfarin to act upon that protein, and that will result in a resistant, right? And then another number B, explain the effect the consequences, the outcomes. Explain the effect of this mutation on humans with thrombosis. We said, let's shift nyana. We said human with thrombosis, they are being affected, their blood is clotting right on the arteries, and then when you eat you drink a warfarin drug, and this drug, it's coming and attached to this blood clot, and then when it attaches to this blood clot is going to be broken down. But if you have this mutation, and then this warfarin, it won't be active, or you are going to be resistant to this treatment. What will be the effect? The effect is that your blood clotting will not reduce, right? Your thrombosis will not be reduced, right? Will not be reduced when you are being treated by a warfarin. And then another thing, because you are saying the effect of this, they're saying 3 marks, the effect of this mutation on humans with thrombosis. Then let's go to the extract. You can say thrombosis, and then you can say that the thrombosin won't be reduced, right? It means that the formation of blood clot in the artery will increase, right? And then you can say the thinning of the blood to break down the clot will not occur, or it will be reduced, since the protein has changed its structure, right? Agree, this drug, it has to come and bind to that protein so that it will break down the blood clot, right? And then if this warfarin is finding that protein has altered, or that protein has changed its structure, it will be difficult for this drug to come and do its work. So this will be the effect of having this mutation. When you have this mutation, you are going to be suffering from this. Will be the results, and the effect will be death, right? You can also write and then come up to T. The effect of this mutation can also result in death, because the arteries, when the arteries they are blocked, the death will come, right? The blood will not be circulating. So that was it from me, guys. 3 marks, it's coming from here. Since the warfarin is used to, some of them they can just come and copy here and say, since the warfarin is used in the treatment of thrombosis, no mark there. Thrombosis results in the formation of blood clot, so this will increase, right? And then foreign causes the thinning of that, and this will be reduced, right? And then the clotting will increase, and then you understand, you can get another 3. So that was where the question was coming from, and the answer we were supposed to get, right? So we also did this. You can agree with me. So that's it from me, guys. Thank you for tuning in. I'm so tired. So thank you for tuning in, guys. Don't forget to subscribe, don't forget to like, don't forget to share, guys. Don't forget to comment, don't forget to reach out on my private WhatsApp number 6168, guys. Don't forget. And then for those who are watching my videos up until the last minute, I want you guys to go and comment hashtag week 3 done. I just want you to comment this, and then when I see your comment, I'll know that this person was watching my video from the beginning up until the last dot, 1 hour 7. You invested your time, guys. Also me, I'm also investing my time to this. I will also appreciate if you are also investing your time to this, right? So thank you. You can see Woma 2.2. You see how there's some questions they can come with 6 marks for only you to describe the process of DNA replication. 6 months. DNA unwind, DNA unzip. You understand? Double the 2 complementary strand will be made. Then your 6 months, you're out. You have to start the beginning. The chromosome will unpacked, and then there the DNA will unwind. Then after DNA unwind, the DNA will unzip. After the DNA unzip, the 2 complementary strand will be replicated. Then after that replication, it's done. I saw somewhere that you don't have to know about the enzymes that are involved in DNA replication, since there are enzymes that are involved in this DNA replication. Enzyme helicase enzyme. So it's fine, guys. Thank you from me. Thank you. That was it from me. I thank you guys. I think until we meet again, we are going to meet on Monday. And I was saying that I have a YouTube, a Wi-Fi here. So if you like, tomorrow we can do a live, we can do Aritsi Baning, we can do a live session where we introduce one another, we talk about our life, you understand what is it that we are coming. Well, now you'll decide the time, guys. Let's decide the time. We are 33 in this class now, 33 subscribers. So that's what I will take it. I'll take it as we have 33 students. So these 33 students, we can have our live on Facebook and we introduce one another. So thank you guys, until we meet again. That was it from me, Mr. Pepo. We'll meet Monday with our topics here, our genetic engineering and what, right? So thank you. Sharp. Peace. Mraro hotso korula ukuthula kube nani.